Hi, Assalamualaikum. My name is Aisyah, Anati binti Johari. So on my part, I will discussing about aim, abstract, and methodology. So for the first one is aim. To determine the evolutionary position of Colponemids, we generate transcriptomes of Colponema vietnamica strain COP7A and two newly discovered relative. To investigate the three main Alveolate lineage are each diverse and important in their own right. And for abstract, alveolates are a large and well-studied group of microbial eukaryotes of significant environmental and ecological diversity, dinoflagellates and ciliates. Alveolate plastids and mitochondria have been particularly well studied because of their functional importance and unique evolutionary histories, and have thus come to play a central role in our understanding of endosymbiosis and organellar evolution in general. Reconstructing the evolutionary history of this character have proved challenging because of the great evolutionary distance between epicomplexan dinoflagellates and ciliates. But one solution has come from identifying early diverging alveolates, the epicomplexan including the causative agent of malaria Plasmodium, the dinoflagellate including harmful algal species and important endosymbionts of coral, which is Alexandrum and Symbiodinium. And the ciliates include the model system for cell biology, which is Tetrahymena and Paramecium, have played a key role in our discovery and understanding of many characteristics that either are unique to eukaryotes or provide a basic for eukaryote-wide concept, including the structure and function of telomerase, catalytic RNAs, and apical complex and cell invasion, permanently condensed chromosome nuclear dualism and DNA rearrangement, and mRNA processing. And for the next one is methodology. We use the report data to describe the measure that has evolved distinct and unusual characteristic and the origin of which have proved to be difficult evolutionary puzzle, which are each diverse and important in their own right, and each includes a number of significant model organism. We also use simulation data to examine how the two lineage illuminate the central states of alveolate mitochondrial genomes. This method allows us to see the mitochondrial genome structure and gene content. So that's all for me. Thank you.
For the introduction, the alveolates comprise three well-studied parties, lineages of significant environmental, medical, and economical importance. These major lineages have evolved distinct and unusual characteristics, the origin of which have proved to be difficult evolutionary puzzle. Mitochondrial genomes are a prime example. All three groups depart from canonical form and content, but in different ways. Colponemids represent at least two independent lineages at the phylum level in multilocus phylogenetic analysis. One sister to epicomplexans and dinoflagellates, and the other at a deeper position. Colponemids are an alveolate lineage whose morphology suggests a possible early diverging position within the group. Colponemid sequences also identify several environmental lineages as Colponemids, altogether suggesting an untapped potential for understanding the origin and evolution of epicomplexan.
Okay, for result and discussion, I have insert three figure, which is figure 1 is 10.4. Colponemids represent two deep branching lineages of alveolates. So let's make it short. Colponemid sequences consistently form two deep and independent alveolate branches which is Colponemid-like Peru, is the closest sister to Myzozoans with strong support, whereas Colponema edophysium and Colponema Vietnam branch together at a deeper position near the base of alveolates or sister to ciliates. So for figure 2, figure 2 is stand for Venn diagram of mitochondrial gene content. So as we can see, comparing the gene content of the Colponemid-like Peru mitochondrion with that of other eukaryotes reveals that it has retained the largest mitochondrial gene set among all alveolates, while Myzozoans contain the smallest and some of the most unusual organized mitochondrial genomes, which encode only 3 protein coding genes and fragments of the ribosomal RNA genes. So, for figure 3, figure 3 is stand for presence or absence of each character is recorded based on the putative ancestral state in the lineage, which we get from the figure 2. So, figure 3 is actually the conclusion that we can get from the figure 2. So that's all for result and conclusion.
Okay, next I will present the conclusion part. The Alveolate complex tree will study British lineage of significant environmental, medical and economical importance. These major lineages have involved distinct and unusual characteristics, the origins of which have proved to be difficult evolutionary puzzles. So, here we describe two such lineages and how they illuminate the ancestral state of Alveolate mitochondrial genomes. We established 5 colonial cultures of Colponemids, predatory Alveolates without cultured representative and molecular data. Colponemids sequence also identified several environmental lineages as Colponemids altogether, suggesting an untapped potential for understanding the origin and the evolution of epicomplexes, dinoflagellates and ciliates. So I think that's all from me. Thank you.